We can see your network.
Thank you for your expert kind assistance. I'm Dr. Saumya Pandey, PhD, recently worked as Head Department of Clinical Research, Indira-IVF Hospital, Udaipur, Lucknow, India, and presenting in the Cancer Pathophysiology session at ICC 2024 with the title for the talk, Toll-like receptors, autophagy, PI3-AKT, immune-metabolic intersections in benign prostate hyperplasia and prostate cancer in the robotic prostatectomy golden era, damage-associated molecular patterns in immunogenic cell death. May I request the slide transition to the next slide, please? A quick cancer research snapshot. Prostate cancer has emerged as a major public health problem worldwide. Prostate tumors shed circulating tumor cells, CTCs, into the bloodstream of patients diagnosed with metastatic, castration-resistant prostate cancer, CRPC, and hormone-refractory prostate cancer, HRPC, and CTC counts predict overall survival and provide an efficient framework for multi-scale analysis of fluid biopsies for quantification, as well as monitoring of prostate cancer in individual patients of diverse ethnicities. Prostate and nearby organs. Prostate cancer forms in tissues of the prostate, which is a gland in the male reproductive system found below the bladder, in front of the rectum. Prostate surrounds the urethra, the tube through which urine flows. A healthy male prostate is about the size of a walnut. If the prostate grows too large, it squeezes the urethra, slowing or stopping the normal flow of urine. Prostate cancer usually occurs in men of a relatively older age. Next slide. Emerging prostate cancer trends. A quick cancer stat fact regarding prostate cancer. The estimated new cancers in 2024 post-global pandemic, coronavirus, Omicron, and the mass vaccination scenario on a global level were 299,010, and the percentage of all new prostate cancer cases worldwide were 14.9%. Estimated deaths in 2024 was 35,250, and the percentage of all cancer deaths peaked at 5.8%. Next. The rate of new cases and deaths per 100,000 prostate cancer. The rate of new cases of prostate cancer was 116.5 per 100,000 men on a yearly basis, while the death rate estimate was 19.0 per 100,000 men per year. These rates were age-adjusted and based on 2017 to 2022 cases and 2018 to 2022 deaths. Now there is a lifetime risk of developing cancer, with approximately 12.8% of men who would be diagnosed with prostate cancer at some point during their lifetime, based on the recent data, excluding 2020 due to the global pandemic COVID, with overwhelming death rates. Prevalence of prostate cancer in 2021, there were estimated 3399229 men living with prostate cancer in United States of America. So life-threatening prostate cancer trends, we all need to join hands for eventual evidence-based predictive and prognostic biomarker development and physiological, pharmacological scaffolds and novel patient-friendly drugs for eventual management of inflammatory cancers ranging from prostate cancer, breast cancer, cervical cancer, glioblastoma, and a myriad of cancers. So we need to dynamically collaborate and join hands for the fight against cancer. Next. Prostate cancer trends, rates of new cases per 100,000 persons by race, ethnicity, all races 116.5, Hispanics 88.1%, non-Hispanic American Indian and Alaskan Native 76.7%, non-Hispanic Asian Pacific Islanders 61.4%, non-Hispanic Blacks have the highest rate of new cases amounting to 188.7%, while the non-Hispanic white 114.9%. This denotes that ethnicity, varying genetic profiles of men have a significant role in prostate cancer development, with the non-Hispanic Blacks showing 188.7 new cases per 100,000 persons by race ethnicity. Next, please. Percentage of new cases by age group of prostate cancer. We see an alarmingly higher rate between the age bracket of 65 to 74. So prostate cancer is most frequently diagnosed among men of relatively older age, with the median age at diagnosis being 67 years. Relative survival is an estimate of the percentage of patients who would be expected to survive the effects of prostate cancer and other cancers. It includes the risk of dying from other causes, because survival statistics are based on statistically powered large groups of ethnically disparate, genetically disparate patient population cohorts. They cannot be used to predict effectively and precisely what will happen to an individual patient. No two patients are entirely alike, and treatment and responses to treatment can vary greatly with different dose responses, different inflammatory and metastasis scenarios. So the relative survival in prostate cancer, with the 5-year relative survival rate based on recent trends, amount to 97.5%, which is indeed an alarming rate. Next. The objectives of my innovative pilot study were to investigate the potential immunomodulatory role of complex autophagy, toll-like receptors, and PI3-AKT signaling networks crosstalks in single-cell immunobiology by targeting circulating tumor cells in benign prostate hyperplasia and prostate cancer, for eventual design of promising patient-friendly, cost-effective predictive and prognostic biomarkers and/or pharmacological scaffolds for future immunotherapeutically potent combinatorial drugs with minimal adverse effects in the robotic prostatectomy era. Cell viability, MTT proliferation, cytotoxicity assays were performed under basal and glucose-deprived metabolic physiological perturbed conditions in TLR4 agonist, lipopolysaccharide endotoxin-treated androgen receptor positive negative prostate cancer cells in culture, primarily the LNCAP and PC3 cells. Autophagy flux was monitored by assessing relative ratios of the autophagy marker microtubule-associated light chain protein 3 LC32 versus LC31 in 0 to 12 to 24 to 48 hours time course in GD-triggered cells in absence and or presence of the TLR4 agonist lipopolysaccharide. This was followed by protein isolation by the radioimmunoprecipitate assay method, RIPA, protein estimation by Bradford's assay, Western blotting with primary antibodies, LC3, Becklin-1, BCL2, ATG25, high mobility group box 1, which is HMGB1, from Cell Signaling Technology, Santa Cruz Biotech, CA, USA. Immunoblots, primary antibody 1:10 ratios were used, was subjected to densitometric scans and relative protein expressions. Fold changes were thereby determined by Kodak imaging software. GAPDH, which is glyceraldehyde 3-phosphate dehydrogenase, and or beta-actin were used as internal controls, and tumor biopsies were collected from clinically diagnosed confirmed patients of prostate cancer with the early versus advanced as per prostate-specific antigen PSA, Gleason grade TNM staging of North American ethnicity, American whites, African Americans, Caucasians, Hispanics from New York State, USA, undergoing radical robotic prostatectomy, and CTCs were evaluated for phenotypic differential expression of target genes involved in autophagy, apoptosis, proliferation, necrosis, using high-throughput precision-based single-cell analysis for the clinical follow-ups of robotic prostatectomy patients, tobacco users, non-users, post-surgery was planned for future biobank development in prostate cancer. And this has tremendous avenues for fertility preservation in prostate cancer with biobanking for prostate cancer in relatively younger patients, despite the alarmingly higher trends of prostate cancer rates in men, especially the African American men of relatively higher age, with the median age being 67 years. Next transition. A quick cancer research snapshot. This is one of my papers, my first scientific publication on immunobiology of toll-like receptors. TLRs are a family of evolutionary conserved pathogen recognition receptors. They have tremendous scope in cancer immunotherapy, in predictive and prognostic biomarker development for eventual evidence-based cancer immunotherapy in ethnically susceptible cohorts of diverse genetic profiles worldwide. TLRs identify molecular motifs of infectious agents, pathogen-associated molecular patterns, and elicit an effective defensive response against them. Mammalian TLRs derive their name from Drosophila atoll, with which they share sequence similarity. There are 10 TLRs that are known in humans, and they follow the MyD88 and the MyD88 TRIF-mediated independent pathways, which is the myeloid differentiation factor 88 and tall interleukin-1 receptor true domain containing adapter-inducing interferon beta TRIF-mediated pathways. These are the key players in TLR-mediated signaling, with fascinating avenues for eventual predictive and prognostic biomarkers in cancer immunotherapy, in prostate cancer, as well as other inflammatory cancers, both of early as well as advanced stages. So targeted manipulation of TLR signaling pathway has immense therapeutic potential for cancer immunotherapy. Next. Different ligands for TLRs 1 to 10. TLR1 has triacyl lipopeptide. TLR2 has lipoprotein, with others like HSP, heat shock protein 60, HSP 70, defenses, PAM3 cis. TLR3 has the double-stranded RNA, poly-IC, endogenous mRNA. And in my study, the trigger was with TLR4 agonist, which is the endotoxin lipopolysaccharide specific for the TLR4. Next. Pathophysiological roles of toll-like receptors in inflammatory diseases, primarily cancers, as well as others like asthma, atherosclerosis, SLE, innate immunity, sepsis, load, rheumatoid arthritis, and inflammatory bowel disease. So the tilting of the normal tissue from the physiological homeostasis, normal physiological milieu, towards the aberrant physiological metabolically perturbed precancerous to cancerous tissue may have the involvement and complex crosstalk and interplay of toll-like receptors with toll-like receptors 1 to toll-like receptors 10. And lipopolysaccharide endotoxin is the agonist for toll-like receptor 4. And lots of studies have been done in cancer immunotherapy with genetic association studies involving toll-like receptors in large sample size based cohorts, even Indian population subsets, focusing on toll-like receptors 2, 3, 4, and 9 in prostate cancer, as well as in HPV-mediated cervical cancer. So lots of thrust and fascinating avenues for future dynamic collaborations in cancer immunotherapy by targeted manipulation of specific biochemical cell signaling pathways with conjugating precision oncology-based high-throughput technology. Next. A schematic depiction of the complex TLR signaling pathway with TLRs 1, 2, 4, 5, 7, 8, 9, and the TLR 3 and 4 with the TRIF-mediated and the MyD88 pathway with eventual induction of pro-inflammatory cytokines and type 1 interferons that have role in upregulation of inflammatory genes in the inflammatory cancerous cell, leading to early and advanced metastatic cancer phenotype. Next. Phosphoinositide 3 kinase PI3K is a kind of lipid kinase that has responsibility of phosphorylating 3-OH group of phosphatidylinositol PI at plasma and cell membranes. The most well-known and studied type of PI3K, which is phosphoinositide 3 kinase, is class 1. That is the ability of producing PIP3 from PIP2, which is the main site for generation of PIP3 is plasma membrane, and it is responsible for recruiting phospholipid effectors such as AKT and PDK1, which are key players of PI3K cell signaling. PIP3 accelerates interaction between PDK1 and AKT to promote phosphorylation and activation of AKT, resulting in cell proliferation, migration, protein synthesis, and cell survival. Furthermore, the phosphorylated AKT has the capacity of activating mammalian target of rapamycin mTOR as downstream target. AKT and PDK1 contain pH plextrin homology domains, which are capable of binding to PIP3. Activation of PDK1 by PIP3 leads to phosphorylation of AKT at threonine 308, and activation of AKT can thereby result by PDK2 at serine 473 residue. Phosphatase and tensin homolog P10 is an oncosuppressor that is able to suppress PI3 AKT signaling, and for this purpose, P10 dephosphorylates PIP3 to PIP2 as the gatekeeper in subsequent downstream signaling. A quick look at the complex PI3 AKT signaling, which is in favor of prostate cancer proliferation and invasion. Upregulation of PI3K AKT promotes viability of prostate tumor cells and prevents apoptosis in these malignant cells. Furthermore, PI3 AKT promotes expression of VEGF, angiogenesis, and the complex interplay leads to cancer progression with anti-apoptotic factors such as XIAP and PI3K-AKT promoting the expression of the anti-apoptotic factors like XIAP while reducing the expression of pro-apoptotic factors in prostate cancer. So lots of complex crosstalk growing between the complex pathways. Circulating tumor cells in prostate cancer management, CTCs in the blood sample collection with the screen cell filtration, CD45 negative selection, single CTC selection using micromanipulator with molecular profiling using biobanking, and further AFM analysis. High content analysis of single circulating tumor cells has the potential to provide a non-invasive means of assessing prostate cancer progression in real time during the course of prostate cancer treatment, and to aid in patient-specific tailor-made evidence-based individualized therapy by monitoring phenotypic, physiological, morphometric, genotypic, and protein expression alterations in real time occurring in response to targeted therapy. Immunoinflammatory role of autophagy in cancer pathophysiology, including prostate cancer. Cellular stress leads to induction of autophagy in metabolically perturbed, physiologically perturbed, glucose-deprived cells by tilting the physiological cell in the normal phenotypic mode towards the aberrant, proliferative, or quiescent, or senescent, even non-programmed or programmed cell death mode, the inflammatory cancer phenotype. So autophagy, key molecules, key genes that are part of the complex autophagy pathway are BCL2, autophagy 1,2, autophagy 5, autophagy 7, microtubule-associated light chain 3, LC32, and LC31, and Becklin-1 is the central player in autophagy, leading to carcinomas, including cervical carcinomas, colorectal cancers, and the autophagy marker LC3 expression has been studied in an exploratory promising study by me in colorectal cancer cells, and similar replicative studies have been performed using the endotoxin lipopolysaccharide LPS, which is the TLR4 agonist in prostate cancer LNCAP cells, with tremendous scope for future design of predictive and prognostic biomarkers in cancer management. Next. Autophagy is a process of cytoplasm and cellular organelle degradation in lysosomes. It has been implicated in homeostasis, and under altered biological metabolic conditions such as cellular stress, the cell may undergo survival and/or cell death. Autophagy may in turn govern tumor metastasis and subsequent carcinogenesis. So, the complex interplay between toll-like receptors, autophagy, and the PI3-AKT-PTEN pathway has tremendous immunotherapeutic potential for eventual design of novel, innovative, patient-friendly, evidence-based cancer immunotherapy regimens in prostate cancer cohorts of diverse ethnicities. Next. Circulating tumor cells in prostate cancer management, highly rearranged copy number variation CNV profiles, and androgen receptor AR-positive phenotypes in distinct CTC subpopulations may precisely predict the therapeutic response using pharmacodynamics and pharmacogenomic strategies in animal models, basically murine models of prostate cancer, and prostate cancer human cohorts of American whites, Hispanics, Caucasians, Africans, Americans, and Asians, including Indian, Korean, Chinese, Japanese ethnicity at the population levels. Recently, genomic changes based on RACGH and limited sequencing have been reported on CTCs isolated with the cell system and the high-definition CTC enumeration method, as well as the nano-Velcro chip assay. Interestingly, CTCs expressing genes related to epithelial mesenchymal transition, EMT, such as Wnt5A, FOXP3, insulin-like growth factor IGF-1/2, epidermal growth factor receptor EGFR, and transforming growth factor TGF-beta3 have been suggested to be strong predictors of metastatic prostate cancer. So tremendous immunotherapeutic thrust on CTCs for eventual evidence-based prostate cancer management and design of prostate cancer immunotherapy regimens. Highly rearranged copy number variation profiles and androgen receptor phenotypes in distinct CTC subpopulations may precisely predict the therapeutic response using pharmacodynamics and pharmacogenomic strategies. I have already discussed the slide. Next transition, please. Accurate isolation characterization of CTCs using high-content single-cell analysis therefore holds tremendous prognostic and predictive therapeutic potential in prostate cancer management in both the American as well as the Asian Indian patient cohorts with different ethnicities and lifestyles. So indeed, single-cell analysis CTC enumeration provides novel therapeutic, clinically relevant intervention points and patient-centric, fascinating opportunities for personalized treatments with targeted inhibitors and combinatorial chemoradiotherapy for specific subsets of prostate cancer patients stratified according to Gleason score, prostate-specific antigen level, and tumor stage metastasis. Next. The percentage of cases and 5-year relative survival by stage at diagnosis of prostate cancer. Localized 69%, regional 13%, distant cancer where has metastasized, with inflammatory genes upregulation with the complex interplay between different cell death and cell proliferation pathways like the TLRs, immunoinflammatory, and autophagy and PI3-AKT. Next. Results and promising data sets. LNCAP and PC3 prostate cancer cells were more than 80% viable under basal and glucose-deprived metabolic physiological conditions in TLR4 agonist LPS endotoxin-stimulated sterile culture in vitro conditions. The autophagy flux was significant in glucose-deprived, triggered starvation and hypoxic conditions with relatively higher expression levels of LC32, which is 14 kilodalton, versus LC31, which is 16 kilodalton in 0 to 12 to 24 to 48 hours time course. TLR4 agonist LPS modulated autophagic flux was significant in LNCAP cells in 48 hours with differential LC32 versus LC31 expression levels. Protein expression patterns of LC32 isoform was significantly higher than LC31 over 0 to 12 to 24 to 48 hours in AR-positive negative prostate cancer cells with significant levels. So hypoxic vascular insufficient necrotic tumor cause were assessed for CTCs and frozen for prostate cancer, oncofertility biobank biorepositories, robotic prostatectomy CTCs, single-cell analysis clinical research data sets in North American patients of New York States, United States of America yielded promising outcomes for predictive and prognostic biomarkers, development for autophagy TLR-PI3-AKT for subgroup stratification of susceptible early versus advanced prostate cancer patients, tobacco users versus non-users with differential survival trends. So the risk ratios, hazard ratios, or the odds ratios post-surgeries. Moreover, necrotopic marker, the high mobility group box 1, HMGB1 protein, apoptotic marker BCL2, and autophagy signature ATG2, 5, 7, ATG10, and Becklin1, which is the central player in autophagic machinery protein expression levels, were relatively higher in TLR agonist GD-triggered prostate cancer cells. Next slide. My promising translational research study strongly highlights the emerging immunotherapeutic potential of autophagy, toll-like receptors, PI3-AKT crosstalk signaling networks in damage-associated molecular patterns-mediated androgen receptor-positive negative prostate cancer for future statistically powered large sample size-based pharmacogenetics, genomics, transcriptomics, metabolomics-based circulating tumor cells, single-cell biology, public health-oriented, meaningful, multicentric, evidence-based, large sample-sized epidemiological studies that may be prospective and or retrospective in ethnically disparate population sets of diverse ethnicities and genetic profiles and lifestyles, tobacco users versus non-users, smokers or chewers of tobacco in states of New York, Texas, United States of America, as well as Asia-Pacific region, North and South India. Further, biobanking in prostate cancer and development of oncofertility biorepositories may prove to be eventually a boon in damage-associated molecular patterns-mediated prostate cancer, infertility, in men's health, urology, oncology, reproductive medicine research globally. Next. Few of my publications. Tobacco as a significant predictor of infertility, a public health perspective in an Indian scenario. This was published recently in Fertility Science and Research, single-cell analysis in circulating tumor cells and prostate cancer, experience of an observership at Cold Spring Harbor Laboratory, Cold Spring Harbor and New York Presbyterian Hospital, Cornell Campus, New York, robotic prostatectomy in urologic surgery, an observership at Yale Medical College of Cornell University, New York, USA, and fertility sterility with emphasis on toll-like receptors and tissue inhibitor of matrix metalloproteases, genetic variants in the management of oncofertility. Next. A recent publication in the field of cancer with thrust on cholangiocarcinomas and gastroenterology and hepatology research for enriching research snapshots in cancer immunotherapy, toll-like receptor and Takeda G protein receptor 5 interplay in immunomodulation of inflammatory colorectal cancer and cholangiocarcinomas. So we may join hands as dynamic collaborators for eventual design of evidence-based toll-like receptors, Takeda G protein receptor 5 mediated, autophagy mediated biochemical, immunotherapeutic cell signaling, target-based approaches for eventual biomarker development or novel drug development in the management of cancer globally. Next. What will the audience learn from Dr. Pandey's ICC 202 for Baltimore, Maryland, USA's online presentation? My promising translational research study offers fascinating public health avenues for the audience today at the prestigious, scientifically enriching, interactive, dynamically engaging, target-driven, and fruitful, meaningful discussion-based ICC 2024 meeting for evidence-based immunotherapeutic regimens by targeting autophagy toll-like receptors, PI3 AKT crosstalk signaling networks in damage-associated molecular patterns mediated AR-positive negative prostate cancer for future large sample size-based pharmacogenetics, genomics, transcriptomics, metabolomics-based CTCs, single-cell biology, public health-oriented, meaningful multicentric large sample size-based genetic epidemiological studies globally with biobanking in prostate cancer and development of oncofertility biorepositories in DAMPS-mediated prostate cancer, infertility, men's health, urology, oncology, reproductive medicine research globally. And recently, a lot of research is going on in the field of artificial intelligence in cancer management and oncofertility. I acknowledge National Institutes of Health, Bethesda, Maryland, USA for previously funded postdoctoral and doctoral biomedical translational research experiences at Schools of Medicine, University of Texas Medical Branch, Galveston, Texas, USA, Creighton University, Omaha, Nebraska, New York Presbyterian Weill Cornell Medical College, New York, USA. And thank you